Hey class, so this is we're going to call this lesson 19. So this is the start of the lecture 15 um, PowerPoint slides. So this is going to wrap up all of the material for exam uh, 2. And so this title of this lecture is Quantitative Genetics. And so in order to understand what that means, we had to look at quantitative traits. So, quantitative traits are traits that have a continuous variation. Um, for example, like in Mendel's world, we talked about purple versus white flowers, but instead of just being purple or white, it could be multiple shades of purple in between. A good example of this is human height. So, uh, if a parent is, 5'6" and another parent is 6'6", doesn't necessarily mean you can always have only have kids that are 6'6" or 5'6". So you could have kids anywhere in between, and so those traits are called quantitative traits. And geneticists can observe these traits and study these traits, and this unit is going to discuss how that's possible. Okay, so the the way that this emerged, the study of quantitative traits, is that uh, these Swedish geneticists were looking at kernel. I don't care that you know their name. They're looking at kernels, and the kernels could be red or white, and so they crossed uh, these individuals, and what they found was this whole range of variation of color. So instead of getting a 9 to 3 to 3 to 1 ratio, they got a 1 to 4 to 6 to 4 to 1 ratio. And that's because multiple genes are involved in this process. This is also how it is with human skin color. And we talked about that, right? There are 3 genes involved in this. And so you have a range of these genes. Let's say in this case, we have each of these puts in some red pigment. And so if you have 4 dominant uh, alleles, then you're going to be dark red. If you only have 3, then you only have 3 units of red. Here there's 2 um, and 1 and none. So you get that entire range of color variation because of they're each contributing some to the phenotypic effect. It's the same thing in height in humans. There's lots of genes that we know of that control height, at least 15 different genes. So I don't really want to get into this, but you can see that there is a variation in quantitative traits. And if we look at, you know, lots of these, you would see a very unusual ratio. So this is the humans that we talked about and skin color. So we know that there's 2 genes, 6 alleles. So it's possible that you could have all 6 of these producing pigment, and then you'd have very dark skin, or none of these producing pigment, and you'd have very white skin. Um, or you could have something in between, right? So maybe you only have 3 of these, so you would have this color. If you have 2, you're this color. Uh, 1, you're this color. It's hard to see on this slide. It's a little bit colored, and then this one is completely white. So we get this range, right? The ratio is 1 to 6 to 15 to 20 to 15 to 6 to 1. And it's possible to have 2 offspring that are white and very dark if you are in between, right? Which is shown here in the F1 generation. All right. So anyway, quantitative trait is described by a distribution um, of individuals in a group. So so if we're looking at height, right, in centimeters, because it's science, um, and then the number of individuals, you would see that there are a lot of individuals that are average, and then some of them are outliers, so they're very short, or some are outliers and they're very tall. So like you could do the same thing for grades in a class. So, lots of people ask me, do I grade on a curve? This is a curve. So, do I grade on that? Um, and so, if we looked at grades, sorry, let me put grades on. No, we're going to put individuals on the y axis and grades on the x axis. And so, let's just go 10, go 10, 20, 30, 40, 50, 60, 70, 80, 90, and 100. So you probably know from grade school that if you look at the back of your report card and you look at grades, C says average. And that's literally what it means. A C is an average grade. A B is good. An A is excellent, and so on. So this is what we're going to focus on, C, because C is average. C is this right here in the middle. What's the average score? And you guys know from experience that a C, it ranges from somewhere between 70 and 79.9%, somewhere there. Um, if you got lower than a 70, you'd get a D. If you got higher than a 79.9, you'd get a B, and so on. So if we're looking at grades, we would expect the average would be in between there, right? Somewhere between 70 and 80, so or 79.99, which would be 70 plus 79.999 repeating divided by. So the average, you just add them together and divide it by 2, and I guarantee you're going to get like 7.4.9999. So we're just going to say 75% is the average. And so it would lie right about here. And we would expect the majority of students to get an average grade. Now, so let's see that this is 40, 30, 20, 10, and 0 on both axes. So we'd expect, let's say, a class of 100, we would expect the majority of people to get 75. There's going to be some people that get higher, but then this number is going to dip down as we go higher and higher. And, so some students are already had genetics, or they paid attention in 181, or they know what's going on, or they studied really hard, or they don't have anything else to do but genetics, and a couple people might hit that 100 mark. And then we're going to have the same thing on the other side. So it's going to be like this. So some people have a hard time. Maybe they didn't get enough information in 181. Maybe their 181 teacher didn't teach them anything in meiosis and mitosis, and they have no idea what's going on in genetics. And or maybe they just don't have time, that life comes up, or something happened with their family, or maybe they don't turn in any work, right? Like that's how my kid is in high school. And so some people are going to get down to here. I mean, most of the tests are multiple choice, so you would expect to get somewhere in, at least if you were just guessing, in this range here. So it's going to range probably somewhere between 20% and 100%. Um, and the average here is going to be 75%. Well, these curves tell us a lot. And to grade on a curve, so let's say that I made up an exam, and that exam, generally I make up exams that are a lot harder than I expect. If I took this exam, and all genetics professor took this exam, the average would be way over here, right? So I would have to curve that average down. How many people would be happy about that? None. Um, but surprisingly and psychologically, if I make the exam harder and the average comes to here, right, then I would curve it up to be online with what the average grade is. So most professors make their exams a little harder. Uh, and that's because they don't really have a way to test the exam. If I made a new exam, who am I going to give it to? The class? That would be silly. Another class? Well, they haven't had genetics. Myself? Well, I would do. It wouldn't be an unbiased survey there. So most professors make their exams a little harder so that they can push it up onto the curve rather than making it easier and pushing it down on the curve. Nobody likes that. I had a physics professor that did that to me at the University of Texas, and I hated the class and I hated him for doing that. So nobody does that, right? Some people do this, and some people, unfortunately, like they don't do a curve. They just put it on at the maximum. So whoever gets the closest to 100, they move the curve based on that. But that's not a real curve, right? This is the curve. The curve here at 60 would be more like this. Let me change colors. So this would be the original curve, and we would move this over. So this is the original curve, and curving the class means fitting it to the original, to the average, expected average, right? Taking the observed and fitting it to the expected average, and not only average but distribution, right? So we're going to look at how is this distributed? What is the steepness of this curve? Is it more like this? Is it more like that? What is it? And so statistically we're going to look at this stuff. Okay. So a lot of times, like we already discussed in skin color, that there is an additive effect, right? You have one. If you have zero alleles, then you're like super white. And if you have one, then you're darker just by 1 unit. Two, you're darker still by 2. And then 3 and 4 and 5 and so this is an additive effect. Every one of these add together, right? So if you have 3 alleles, you do 1, 2, 3. You add 1 and 1 and 1. If you have 5 alleles, you're going to add 1 plus 1 plus 1 plus 1 plus 1. And that gives you 5. So there's additive effects. We ought to also have to take into account things like dominance. So is the trait dominant or recessive? Because if it's dominant, then it might mask that expected, quantitative range, right? Maybe uh, allele number 2 in human skin overpowers allele number 1. We could have epistatic effects, right? So they could be in a pathway. So rather than just additive, we talked about this, so we have A to make B to make C to make D, and then that's our final product. Uh, and we know that that would give us a weird ratio. All of these things can contribute to the phenotype, what we observe of a quantitative trait, right? And this could be anything. This could be, egg size in chickens, or wing size in chickens, or beef on a cow, or crops in a field, or the size of strawberries, or whatever. There's always going to be a range in nature. So that's what we're talking about, quantitative effects. And a lot of times geneticists need to know that, right? If I want to make bigger strawberries, I would need to know, well, what causes the size to vary in strawberries? Is it epistasis? Is it dominance? Is it an additive effect? Is it the environment, right? Like if I had 2 turtles, one I fed and one I didn't feed, and one grew big and one didn't, then that's an environmental factor, because let's say they're in the same clutch, which means basically they're siblings, they're brothers and sisters. Then they would have similar genetic traits. So there shouldn't be a lot of variation in them unless it's environmental, right? If you had a clone, it would be even better, right? Like you had identical twins. So a lot of studies are done on identical siblings to find out. And, believe it or not, there's twins in like, the animal kingdom too. So you have twin chickens. And so maybe you take one chicken, chicken one, and you feed it and see how much of this is additive and dominance and whatever, and chicken two, and you feed it, and then you want to tease out what variation is due to the environment and what variation is due to genetics, and that equals your phenotypic variation. So, and this genetic variation can be additive, dominance, epistatic, whatever. Anything that's genetic goes in here. So, whatever variation is caused by the environment, plus whatever variation is caused by genetics, and we can break this up into VA and VD and VE. Well, that's a bad one. Let's trying to figure out. Let's just say epistasis because we already have E for environment, and then this P is for phenotype. Okay. All right. So we just talked about this. It could be due to non-genetic factors like the environment. If I feed one chicken more, uh, maybe it grows bigger. Maybe it doesn't matter. Uh, so if you're a farmer, you'd want to know if I breed this chicken, this big chicken, to this other big chicken, will I have a big chicken? Because if it doesn't matter, then you just let the chickens run around and breed with each other, and who would care? You wouldn't need to selectively breed them. So nutrition is important, right? Fertilizer, it could be the pH of the soil. It could be, and like I said, we can study this using identical twins because we know their genetic genotype, their genetics is exactly the same. Okay, so why are traits continuous? So usually this involves lots of genes, polygenic, right? We talked about that in human skin, and variations due to the environment. So when we look at all this stuff, like let's say we have this is the mean, and this is the mean, and this is the, and we look them all together, we're going to get this continuous uh, variation in a population, and it's going to look like a standard bell-shaped curve, just like we discussed previously. Um, and then this is just showing you we're going to kind of use this model here um, for the class, but it's the same thing as this, right? We're creating a bell curve. So here's just not, it's not as pronounced because there's not as many individuals. But here's our bell curve, right? This is going to be a little flatter. It's still going to be a curve, and then we have another bell curve, and so on. So here's the question: we have two uh, individual bell curves, and so in quantitative traits, so let's say that this is a short person. What is this percentage of plants and crawling? Okay, so anyway, let's just say that's a short person. This is a tall person, and they interbreed. You would expect their offspring to be in an intermediate range, um, and then if we do the F2 generation, so that means that siblings are mating with siblings, then we're going to get a broader range. And then if we go to the F3 generation, so we breed these together, then we're going to get a discrete, narrower, discrete range of two populations, right? And then if we continue on, we're going to get another discrete range and another discrete range. So in F3, we're going to get all these variations. Some are going to overlap and some aren't. But this is how we study quantitative traits. Again, this is to zoom in on that graph. And like I said, this is what we're going to kind of focus. So this is our quantitative traits. We're not looking at a lot of individuals. So this is the percent. Um, and then we're going to get in F1, we're going to get a more narrow. So these are going to sort of come together. It's going to look similar, kind of like these are combined, so they're averaged. And then when we do a cross between two individuals, uh, they're going to be purebred. And to do that, and this is one of your assignments, is if things stop outbreeding and they start inbreeding, what happens to their genetics? So you just look at each generation, right? Look at the parental generation. What's the percent of each of the alleles? And then in the F1 generation, right, there's going to be a different percent. That percent is going to be based on what the probability of what's getting passed on. And then F2. And if you inbreed things, the eventually you're going to get them to the point where their genetics are all identical. That's what geneticists do. And so I wanted you to look at this because I think I have a problem where you have either one or two generations of inbreeding, and I want you to see what happens to the alleles whenever you do that. And you'll see for yourself that it reduces uh, heterozygosity and goes in this direction, right? So that we have alleles that are fixed in the population. Eventually, one of these will get taken out, and you'll be left with, if you kept backbreeding individuals, you would be left with one genotype after about 20 generations. And so this is how scientists make pure breeding, by just backbreeding them to the original parent over and over. All right. So anyway, there's a difference between the parental lines due to differences in alleles at multiple loci. So again, that's polygenic. We already talked about that. All right. So I just put these in here so you could see. Let's say that this is homozygous for A, and we're just going to use subunits 1 and 2. Homozygous for B, homozygous for C. So we're going to see A1, A1, B1, B1, C1, C1. And this is homozygous for the other allele, right? So this is going to make it taller. Remember, it's an additive effect, so we're going to get taller. And then in here, we're going to get an average in between those two, right? Additive effect. So we should only get 3 units of tallness, where this might be 0 and this might be 6. Now we get 3. And then if we go through to the F2 generation, you'll see that there's going to be more variation, right? 27 possible genotypes. You could do it out. Do the Punnett square if you want. And you'll see. That's why I just put a dot, dot, dot here. And then once you do that, if we, let's say we inbreed these, and we breed this group to each other, then we're going to get this population. If we inbreed this group to each other, we'll get this population. So let's say we're doing chicken size. So in this case, we know that if we pick big chickens, we breed them together, we're going to get offspring that are big chickens. And if we have small chickens and breed them together, we're going to have offspring that are small chickens. And if we take some from here, they're going to be in this range. And if we take some from here, they're going to be in this range. Okay. So how do we statistically analyze these quantitative traits? So how do we predict what these are going to be? And so there's 2 primary statistical methods used here. One is the mean and one is the variance. We also look at standard deviation. The reason is because it's in the same units as the mean, whereas variance is not. And you'll see that in a second. Okay, so this has the same mean, right? Right here. But the variance here is different. So that widens. This is more variable in this population than, say, this population. there's more range. This has a different mean, but the same variance, right? So these hills look exactly identical. They're just at a different, their peak is at a different point. And then this is different means and different variances. So this would be this mean. This is a very steep variance. There's not a lot of variability in this population. This is a different mean. This is a very shallow hill, so there's lots of variation in this population. And you could get even more, like so. Anyway, so how do we calculate this? It's really not that difficult. I'm sure everybody has calculated the mean, which is it's the same word as average. That's a V in case you didn't know. And so all we're going to do is to get the mean of a population, we just add up all the individuals in that population and divide it by the number of there are. So let's say we have a population that's 30 and 40. Let's just say that this is shoe size. I don't even know if this is accurate or unrealistic. Let's see, 2.54 centimeters. Okay, I did the math real quick, and this might be a little unrealistic. Like, this is probably a size 11, and this is a size probably 16. So, if you're going to do in this, shoe size is generally in inches, although there's some variation between women's and men's sizes. Women's, you might look at subtract an inch, men's, you might want to add an inch. Anyway, so 11 to 16 inches is what this range is. Um, and so let's maybe this is for basketball teams. So so we do like 32 and 34. And so if we want to get the average, we just add them all up. So 30 plus 40 plus 32 plus 34, and then we divide it by the number of entries, which is 4. And let's see what this is. Where's my mouse? Oh, okay. So this ends up being 136 divided by 4 is 34 centimeters. So this is the average or the mean, which is the symbol for mean of this set, right? These 4 individuals with their giant shoe size. Okay, so anyway, and then uh, variance is the average of the squared differences from the mean. And then standard deviation is the square root of the variance. Um, that gets it back into the same units because we're squaring this. So if we want to get it back to units that are related to the mean, we have to square root it. And I'm not going to get into this equation right now, but I'll show you. It's not that complicated. It looks complicated, but it's not. Okay. So let's look at dogs. Let's say we look at the shoulder heights of dogs and they're 600, 470, 170, 430, and 300 millimeters. And so the arrows are showing where we're measuring them to, their shoulder height. So the mean is all of them added together and we divide them by the number of dogs. So 1, 2, 3, 4, 5 dogs. Our mean is 394 millimeters. So the average height is 394 millimeters. And we would say the mean of the phenotype is what MP stands for, but we could just use the same symbol, mean. It means the same thing. No pun intended. So for variance, we want to look at the variability in the population. So for variance, this symbol is standard deviation. And if you've taken a math class, you probably know it. And so the variance, sometimes written as V, but it's also sometimes written as standard deviation squared, because that's what it is, literally. Um, we're taking the square of the deviations. So we square each of the numbers, right? So here, the question is, here's our mean, right? We just calculated our mean, 394. So our mean is 394, which is symbolized right here by this green line. Now we want to know how much do these dogs vary from that 394. So this dog here is, what do we say, 600? So that dog is at 600. And so in order to figure out how far it is from the means, we take 600 and subtract 394. And we get 206. That's where this number came from, and we square it. And then we just go down the line. So this dog, Whippet, whatever it is, Greyhound, this one, I forgot, what's it measure? 470. So we take 470 and subtract 394, and that gives us 76, and then we square it. And then we do the same. So this one's a negative value. What is that? Wiener dog. 170. So we take 170 minus 394, and that gives us a negative 224. And then we square it, and so on and so on. And then we have 5 dogs, right? So we're going to divide that by 5. If we do the math, 206 squared is 42,436. 76 squared is 5,776. Negative 224 squared is, 50,196. 36 squared is 1296. Negative 94 squared is 8836. We divide them all by 5. So this is the total. We're going to, we're summing them all together. 108,520 divided by 5. So the variance, the variability in the population is 21,704, which is the square of the difference from the mean. All right, so to get it back into something meaningful, because that's a huge number, we square root the variance of the phenotype. So we take 21,704, we square root it, we get 147. So the standard deviation, this is standard deviation. Remember, this is variance. And so our standard deviation is 147. That kind of gives you an idea. Like, these dogs vary plus or minus 147. What do we measure them in? Centimeters? Millimeters. So they vary about 147 plus or minus 147 millimeters on average. And so we get our curve that way, right? We can look at our curve. The standard deviation would be this if we're on the same unit as the mean. And so you can see why that would be a little more useful. Okay, so let's do some real world problems. Let's say that we want to calculate the mean, variance, and standard deviation of the body length of 10 of spotted salamanders from whatever Penobscot County, Maine. All right, so we go out and we collect a bunch of salamanders and we measure their body length in millimeters, and this is what we get. And so you know that in order to get the mean, we add them all together and then we divide it by the total. So there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And so if we add all these numbers up, the sum of this values is 571. I know there's a lot of crazy math formula things in here, but this is just saying this means sum, right? The addition of all of this column. And then it's just saying x, right, is just a variable, and then this I is just designated as uh, the original body length, right? So that's just that variable. X, it is just designating it as I versus X, uh, II, or whatever. Just like alleles, right? So we put allele, 1, A1, A2, A3, and so on. Okay, so we take 571, we divide it by n, the total number of salamanders, and this is our mean, right? This is the mean. Now we want to calculate variance. So we have to look at every one of these. We know the mean is 57.1, so we have to subtract it from every one of the body lengths, and this is the values that we get, right? And so we know that we have to take that and square it. We have to square every one of the differences from the mean. This is the values that we're getting. Right? So this is just taking body length and the mean, which is what this X represents here with the line over it. You can hardly see it, but that's what it is. This minus the mean, right, is this. This minus the mean squared is this. We need to do that to get variance. We add them all together. We get 112.9. This is the sum of body length minus the average body length, right? That's what the X over hat means. We square that to get variance. So we get 112.9. These are all squared values. We divide it by 9. Now, this is weird, right? Like I just told you we divided by 10. And the truth is that that would be a standard variance. But because we're not doing the entire population, we're only doing a subset of the population, we have to take that number and subtract 1. So n minus 1 is for a subset of a population. We didn't collect all the salamanders in Penobscot County, Maine. So if n is the total population. All right, so anyway, this is why we have to subtract it from 1. So we get 9 because 10 minus 1 is 9. And this is our variance. So now this is the variance of a subset of population. We're only looking at 10 out of the whole population. So this is a subset of the population. And then the standard deviation, which is S of X, right? This is the body length. The standard deviation of this variance is the square root of the variance. So in this case, we square root this. And so that gives us 3.54, which is our standard deviation. Okay, so on the exam, I expect you guys to know this stuff. I'll put in the formulas, but you need to know what they represent. All right, covariance is if we look at two traits, how are they associated with each other, right? Like, if someone's taller, would you expect their foot to be bigger? Is there association between foot size and height? And so that's what we would say is they're covariable. They vary together. They could go opposite directions, right? Like, let's say you're, I don't know. Let's say you get taller. I'm just making this up. So let's say height is inversely correlated or covariable with toe size. So let's say your little toe, the taller you are, the shorter your little toe is. That would be a covariance of different directions. Whereas if we say your height is, the taller you are, the bigger your foot size is, then those would be covariable. So anyway, if they show a similar behavior, then the covariance is positive, right? This would be a positive covariance. If the traits show opposite behavior, then the covariance is negative. So for this case, this would be a negative covariance. This would be a positive covariance. If the covariance is 0, then that means the traits are independent. So maybe we look at hair color and height. And so maybe it's all over the place. And so, if we were going to graph it out, it would be a flat line. It wouldn't matter. Uh, let's say this is your how tall you are and number. Let me get a more highly variable trait. Let's say how tall you are and the color of your skin. All right. So let's say it doesn't matter. Skin color has nothing to do with your height. Maybe it does. I don't know. I haven't studied that. But that would be a covariance, right? So let's say uh, we could covary like sexes. So let's say your mom's height and your dad's height. And so are these related to one another? Well, if that were true, and dad had a kid, let's say this dad is 6'6". I don't know what that is in metric system. We're just going to go back to the imperial system. Um, and let's say mom is, she's also equally tall. She's 6'6". Well, if it wasn't covariable, then this wouldn't depend on this. So maybe the kid is, uh, I don't know, somewhere in the between. So if mom and dad's height were both contributing, then you'd get a line, and that would be a positive line. If it was inversely proportional, so the taller mom is, the shorter the kid is, taller dad is, the taller the kid is, then this would be a negative slope, right? And that's kind of what covariance. And if it doesn't matter, it would be a flat line, and the slope would be 0. So covariance is kind of the slope of the line, um, and to see if two traits are variable together or not. Does it matter or does it matter? This can be important to farmers. Maybe you want to get big chickens or big eggs, or you want heavier eggs, or who knows? Maybe they pay more for pumpkin seeds that weigh more. So you might want to breed pumpkin seeds that are heavier versus ones that are lighter. I don't know. I'm not a farmer, but I know people, this is super important to their livelihood, and so that they come to geneticists to figure this out. All right. So let's say we want to see if there's any correlation between body length and the width of their head. So we're going to have two variables here. We have x. We're going to let x be the body length, and y is going to be head width. And so we're going to say the covariance of x and y equals the sum of x times y minus 1 divided by n. Remember, because we're doing a subset of the population. This is why we do n minus 1. And then we're going to multiply that by the sum of all the x's and all the y's. And that's going to give us the values. So we got to sum x and we got to sum y. We know the number of individuals. We add up all the x and y values, and then we put them in the equation. So here's a data set. And basically, this is just the same thing they were doing before. We're just taking the difference between x and y. All right, so we have, dang it. So we have x. We're just going to call this I. So here's our body length. We add them all up, right? We sum them. This is our body length. We divide that by the number. So there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 salamanders in this population. So the sum of all the body lengths divided by 12 gives us 79.92. Then we're going to do the same thing with y, right? So here's the head width. This is the variable head width. So we add it all up. There are, again, 12 individuals. This is the head width average in millimeters. So 213 is the sum of all the y's. Divide that by 12, and then this is the average or mean of y. So we have the average, we have the mean of x, and we have the mean of y. Right? That's what that line over the top means. This is the average. So the variance of x is that value squared. So we take the sum of xi minus x squared divided by n minus 1. So this is exactly what we did before. All right, we're going to take the difference between the number and subtract it from the mean. So if you did the math out here, this would be 72 minus 79.92, which is 7.92. And so we square that, right? What's the square of 79.92? Should be 62.67. Let me just check that, double check that. 7.92 squared. It is indeed 62.7264 is what I get. So obviously they're truncating something here. They're rounding it. But anyway, that's close enough. So we add these all together. So this is the sum of the difference, and this is our value. This is exactly how we calculated variance before. So here's our variance, and then we had to divide it by n, right? Remember, n minus 1, because this is not every tiger salamander on Earth's length and body length and head width. It's a subset, and that's why we subtract 1. And so and that's what you do scientifically to account for variability in a small set. All right, so 153.35 is what we get for the variance. And then, of course, to get the standard deviation, we square root that, 153.35, which we already talked about. And that gives us the standard deviation of 12.38. So that's our standard deviation. Now we're going to do the same thing, get the variance of y. So we add them all up. We take the mean, subtract it from the actual value, and then so we get y1 minus the mean. These are the values that we get. We square them, right, to get the variance, and this is what we end up with the variance. So 94.25, sorry, that's the sum of all of these values. We have to divide that by n minus 1, so there's 12 individuals minus 1, which would be 11. So we divide it by 11, and that gives us 8.57. All right, so we have the standard deviation. We square root this, square root 8.57. That gives us 2.93. Now we got to do the covariance. So to do the covariance, we're taking the sum, right, of these multiplied together. So 72, come on. 72 times 17 should give us 12,224. So let me just check this. Something going on with my neck. 72 times 17 equals 12,024. That's correct. So we multiply them together. We get all these values. We add them all together. That's what we're doing. And then we should get a total of 17,385. That's what this value is here. Now we're going to subtract it by 1 divided by 12, right? That's the number of individuals in. And then we're going to multiply the sum of all of our body lengths, which is 959, which is right here, and the sum of all of our head widths, which is 213 right here. And then we divide that by 12 minus 1, right? So this is n, where we subtract 1 because this is an all-inclusive population. And we end up with a covariance of 32.97. Remember, this is a squared value, so we want to get it back to a correlation coefficient. Like, how are they correlated to each other? In other words, what's the slope of the line? So the correlation coefficient, we take that covariance value, which is here, and we divide it by the standard deviation of x and the standard deviation of y multiplied together. And if you do the math, you get 0.91. So that would be a positive line with a slope value, y equals mx plus b, standard slope value. This slope would be 0.91. So we would expect almost a 1-to-1 ratio. So as the cell, this is like head size This is body size. And so there's a very strong correlation. As head size increases, so does body size, right? And that's the slope of our line. That's what correlation coefficients are. So on the test, I might ask you to, I probably won't, but I could. Because these values are very important geneticists to figure out um, how traits are related to one another, right? Is chicken size related to the size of a chicken egg? You might want to know that if you're trying to make, extra large eggs. Okay, so this is just an explanation of what we just talked about and how we came up with a correlation coefficient. So there is a thing called regression. So I'm just going to talk briefly on regression. I don't really want to get into it, but uh, when we're doing covariance, we're looking at two variables, right? Like parents' height, mom's height, and dad height. We're doing regression. We're keeping one variable constant, so there's an independent and a dependent variable, and so this gets down to heritability. Um, so let's see. So we want to know what is heritable, right? We have a genetics component and we have a phenotypic component. Maybe we feed the chickens more and they get bigger, or maybe they're just born genetically predisposed to be bigger. Um, so if we let P be the phenotype in this model here, this is our phenotype, and this is the maternal component. This is the paternal, so mom and dad, plus environmental factors. This is the a simple model of how we can have additive genetic effects. Um, so we have the contribution that mom makes to the phenotype, plus the contribution that dad makes to the phenotype, plus the environmental factors, gives us the phenotype, right? So this is total phenotypic effect from mom, total phenotypic effect from dad, and then phenotypic effect that's not due to anything that's genetic. It's simply due to mom or dad's genetics. All right. Let's see. I mean, if there's a large variation in the population of, say, mom, let's say height or whatever we're talking about, and dad, then there's going to be a large variance in the population. Different individuals can vary between different environments. So, a lot of studies of this of variance is looking at genetically identical individuals, because if we do that, then we can see what if we assume the genetics is the same and we put them in different environments. So these are generally individuals that are separated at birth and raised in different environments. We can look at what the phenotypic effect is on things that are solely environmental and nothing to do with genetics, because their genetics are identical, right? So any variation you see between twins has to be variation that's experienced due to the environment, which is this E value here in this equation. All right. So I'm not going to go through all this math. We just figured out how to do the mean and the variance, so I'm not going to go over that. So if we calculate the variance of the phenotype, right, and I showed you how to do that with we did salamander length and then head length and body, whatever it was, and we look, so we can calculate genetic variation, the variation due to the variance, right, the variation due to mom, uh, variation due to mom plus the variation due to dad, paternal, right? You could just I'm just going to use D. So the variation from mom and the variation due to dad added together is all the genetic variation that you're going to get because no one else is contributing to your genes. And then we can add that to the variance due to environmental stuff. So this is the equation, right? Your variance of your phenotype is due, how much this varies is due to the variance from mom and dad added together plus the variance due to the environment. Easy enough. There's the equation. Not hard. Okay. So, but there are we can sub divide out these things due to the variance of genetics. And we talked about this, right? So we talked about we can have additive effects like for skin color. So we can say VG, the variance due to genetics, is caused by the addition of additive variance and the addition of let's see, oh, it's gonna go away. Dominance. We talked about dominance, so we have VG equals the variance due to additive effects plus the variance due to dominant effects. And then we don't use E because E is environmental, so we use I for epistasis. So in the end, we get VG equals the additive variance plus the dominance variance plus the variance due to epistasis. Remember, I is what this is dumb, but I mean, you could call this EP if you want. I don't really care as long as you come up with the right answers. But geneticists use I for epistatic variance. All right, so this is just what we drew out, right? So we have the phenotypic variance, the genetic variance, and the environmental variance. So here's the genetic invariance. We can put all this into this category, and this is our end equation. Okay, so we have additive, dominance, epistasis, environment, and then there's another one called G, which is variation associated with genetic environmental factors interacting with one another, which is really complicated. So I don't even know why I brought that up. We're going to get rid of that one. In the end, that tells us our phenotypic variation. So for the test, you guys will have to plug these numbers in to figure it out. So let's just do an example, right? Let's do a real world example of looking at where's my okay, there it is. Variance of the phenotype due to additive and dominance effects. And then we'll look at wings and birds, wing size and birds. So this might be a little easier to understand this. So here's our dominance effects. And let's say if you're heterozygous, this is also um, anyway, so let's say you're big A, right? The dominance is that you increase your wingspan of two, what are these units? Centimeters, right? So let's say we have a bird. Well, if it's big A, then I don't know. I don't know what the average wingspan. Let's just say it's 19, just for fun, because that's what it looks like. So 19 centimeters. If you're big A, you get to add 2 centimeters. So that would make you 21 centimeters. Because it's completely dominant, it doesn't matter if you're big A or big A little a. If you're heterozygous or homozygous dominant, you still get to add 2, right? So this or this equals plus 2. That's dominance effect. Little a is 0. So it's either all, right, complete dominance, or nothing. Additive effects are, they add together, right? So if you have big B, you get a plus 2. Big B, you get a plus 1. Little b, you get a plus 0. So in the end, let's see, you get a plus 2 for this and a plus 1 for this. So this is probably not accurate. We're going to say this would be 19, 18, 17, 16. So the average is probably 16. We get to add 2 because we're big A, little a, right? Dominance. And then we get to add, so this is big A, little a. And then we get to add 1 because we're big B, sorry. This should be a 1 because we're big B, little b, right? So this is the variance of additive. This is the variance in the population. This is the variance of dominance plus the variance of additive, right? We're adding these together. So this is the variance in the population. We're adding 2, we're adding 1, and we end up with a wingspan of 19 centimeters. Same thing here. So if our variance is 16 and we have big A, and big B, we get to add 2. And we get to add, so big A gives us plus 2. Big B also gives us plus 2 right there. So that would give us a score of 20 here. Then this one is 2 plus 1 is we get a plus 3, so that gives us a 19. And so on and so forth. And if you get a plus 0, then, and we get a 1 to 2 to 1 to 2 to 4 to 2 to 1 to 2 to 1 ratio if we look that out. So this is an example of how we could have variable things due to dominance, variance due to dominance, and variance due to additive effects, contributing to our genetic variance. Hopefully that makes sense. So we're not going to calculate the values here. I'm not going to show you how to calculate VA and VD, except for rearranging the equation. So in this previous thing, if we did the math, we would find out that VA is 1, right? The additive variance is 1, and the variance due to dominance is 0.333. Again, I would give you these values. I would not expect you to calculate these values from a data set. Um, so here's an example of like a problem that I might give you, right? So I might say, okay, what's the phenotypic variance if the environmental variance is 0? Remember, you don't have to calculate this. I'll give you these values. The additive variance is 1, and the dominance variance is 0.333, which is what we did with the bird wingspan. And so in the end, we have nothing involved in environmental. The additive value is 1. The dominance value is 0.33. 1 plus 0.33 is 1.3. So our phenotypic variance is 1.333. Okay, so let's say we want to know if it's all environmental variance, right? So if we want to do that, then we have to have zero genetic, VG has to equal 0. There has to be no variance due to genetic factors. So let's say that we have a clonal strain, right? That means one individual is a clone of each other. Same thing, it means exactly the same as identical twins. So at the end, our phenotypic variance is going to be equal to our genotypic variance plus our environmental. So our phenotypic variance is 0 for VG, right? There's no variance. Um, it can vary, but it doesn't mean it could be to soil conditions, moisture, temperature, light, whatever. But whatever it is, it's not genetic. So we assign this as a value of 0. VG equals 0. So we're plugging this in. This value is 0. So VP equals 0 plus the variance due to the environment. Everything is due to the environment. Um, okay, so here's a sample problem, and there are some problems like this in um, your problem sets. So genetic variance is additive and dominance. So let's say I give you that genetic variance is 605, and I want you to solve for the additive variance. I give you the D, so we get the dominance variance. So we get 605 equals, right? This is all the genetics. So VA plus VD plus VI plus whatever. Um, Everything that has to do with genetics. So that is VA plus, and I give you the D, which is 205. So I gave it to you in the problem, right? I said mean weight of the pumpkin seed is 100 milligrams. Um, a supplier of pumpkin seeds wants to pay double for your seeds if they weigh 125 milligrams. So your goal is to get the 125. The more 125s you have, the more money you're going to make. So you're going to select seeds that average 140 milligrams as parents for a selective feeding program. This is the values that I've given you. The environmental variance is 95. Genetic variance is 605. Again, you won't have to calculate this. This will be given to you. Dominance variance is 205. So how do we figure this out? So first of all, first question is what's the additive genetic variance of this trait? Well, we know it's VA plus VG. Environment doesn't have anything to do with genetics, so we don't even include that. Rearranging this, we get VA equals, right? We just divide it out. Um, Actually, we don't do that. We subtract it. So we subtract, so this is plus 205. We subtract 205 from each side. That's 0. 605 minus 205 equals VA. And so in the end, we get 400. VA equals 400. Sorry. Okay. Okay. So when we're doing quantitative genetics, one of the things that we want to know is what factors of an individual's phenotype is due to genetics and what factors is due to the environment. And so quantitative genetics have figured out mathematically how to do the what we call the broad sense heritability, which is defined as the proportion of the phenotypic variance that is due to genetic differences among individuals in a population. So we want to broad sense heritability is looking at the variance in a population that's due to genetics um, among individuals in that population. Um, so, what is due to genetics and what is due to non genetic environmental factors? That value is broad sense heritability. So, if we write it out, it's written as a capital H squared. That's the variation of genetics divided by the variation in the phenotype. Remember, the variation in the phenotype is simply the genetic variation plus the environmental variation. So that's broad sense heritability. We can use this to determine phenotypic correlations of relatives, the response of phenotypes to natural selection is determined by this broad sense heritability, and this can change over time. All right, let's go to our problem and let's calculate a real world broad sense heritability. So we have broad sense heritability is VG over VP. We know from the problem that our VE is 95, our VG is 605, and our dominance variance is 205. So the phenotypic variation is the variation due to genetics, right? Which were given as 605, um, plus environmental, which were given it as 95. So 605 plus 95 is 700. And then so we take, remember, broad sense heritability is VG, which is 605. And then the variance due to the phenotype, which is the genetic plus the environmental, which is 700. And then we get our broad sense heritability is 0.86. That would be a positive correlation, right? With a slope of 0.86. So, what may even be more useful is knowing what the proportion of the variation found in the population is due to the additive genetic effects, right? And so that is narrow sense heritability. So we're all we're looking at is the additive effects of a population, which is VA. So narrow sense, which is small h squared, is the variance of additive effects divided by the phenotypic variance that we see. So this is the proportion of variability that's passed on from parent to offspring, right? Because they're going to be additive. Where complete dominance or non-dominance, like recessiveness, you're not going to see that, right? You're not going to see that in the heterozygote, but you're going to see the additive effect just like we did in human skin color. So again, we're correlating the this is the majority of it, additive genetic variance in the phenotypic variation. So when H is 0, let's say this is height of mom and height of dad, and we have the offspring. They're all over the place, so we can't really draw a straight line. So that would be 0. Our slope is 0. If they're directly correlated, it would be 1. And if they're inversely correlated, that would be -1. And we could have any thing in between. So when narrow sense heritability is 1, all the variation is due to heritable genetic differences. Right? If it's 0, then none of the phenotypic variance is due to genetic differences. In other words, genetics doesn't matter. It doesn't matter if you take a big chicken and breed it with another big chicken, the size of their offspring isn't going to be affected by the size of the parents. So that value H2, squared, um, is 0. If it does matter, if we take a big chicken and breed it to another big chicken, we would expect big chicken offspring. And if we take little chickens and breed them to little chickens, we would expect little chickens offspring. And so there's a direct 100%, 100% correlation between size of the mom chicken and the dad chicken and on the size of the offspring chicken. So that would be narrow sense heritability equals 1. All right. Uh, this doesn't really matter. Like, I think I've explained it well enough. So if it's greater than 0, right, then it's heritable, right? If this line, come on, where's your pin? There we are. If this line is slopes at all in either direction, then it's heritable. If it's flat, then it's not. If we look at the parents and we look at the mean, right? So let's say we take this group, and we get the same ones, or this group, we get, this curve. If it's not heritable, then they would all be in the mean, right? It wouldn't. So in this case, we have small chickens and small chickens. We're going to get an average of small chickens. Big chickens and big chickens, we're going to get an average of big chickens, right? If it doesn't matter, then we're going to get the same average that we would always expect, right in the middle of our population. Okay. Uh, Okay, I'm gonna stop here. That's an hour and 15 minutes. And then I'll finish this up in the next lecture, and then you guys will have all of the stuff you need to do um, your problem sets. Okay, so we're gonna stop at slide 46. This is covered 1 through 46, and then I'll pick up 47 in the next lecture.